Our last and final presentation for the 2023 MSRGN Genetics Summit is entitled Data-Driven Approaches to Define Rare Disease. Our presenter is Dr. Melissa Haendel, Chief Research Informatics Officer at the University of Colorado Anschutz Medical Campus. Unfortunately, Dr. Haendel had an unavoidable conflict arise and is unable to be with us today. She graciously, however, prepared and recorded her presentation for us. Please reference your screen for CEU accreditation and disclosure information. You may also reference the Continuing Education tab in the attendee website for this information. Dr. Haendel has disclosures as noted on this slide, and which she also references in her presentation. We will now begin Dr. Haendel's pre-recorded presentation. We hope you enjoy it. Thank you.
Hi, everyone. It's my great pleasure to be here today to talk about data-driven approaches to defining rare diseases. My name is Melissa Haendel, and I'm from the University of Colorado Anschutz Medical Campus. And these slides can be found at the following bit.ly, Haendel-MSRGN-2023. And some of the content that I'll be presenting today is also found at the publicly accessible website, monarchinitiative.org. So learning objectives for today are to learn how to use the Mondo disease ontology as a rare disease coding system and to understand semantic phenotyping to aid rare disease diagnosis. I have a disclosure to make, which is that I am a founding partner of the Alamaya Health. It's a genomic diagnostics company, and no content that I'll be presenting today has been developed by Alamaya Health. So 1 in 10 Americans, roughly 400 million globally, are affected by rare diseases, and each patient's characteristics are often thought to be akin to the variation in zebra stripes, which is why the zebra is often a symbol of rare diseases. But however, when you hear hoofbeats, think horses, not zebras, because the reality is that there are over 7,000 rare diseases as per the Orphan Drug Act in 1983. However, as I'll show you soon, this number is demonstrably wrong. And obviously, since 1983, we've learned a lot of biology and clinical medicine since that number was captured in 1983. So the criteria for what constitutes a rare disease varies around the world. In the Orphan Drug Act, it was stated that a rare disease affects fewer than 200,000 people. In 2000, the European Union considered the disease to be rare when it affects fewer than 1 in 2,000 people. And these definitions vary in other countries as well. So new diseases are discovered all of the time, but the number really hasn't been updated since 1983. New rare diseases are discovered every week by organizations such as the Undiagnosed Disease Network, the literature, the online Mendelian Inheritance in Man, et cetera, have new weekly entries. And of course, N of ones are matched, defining new diseases in systems like the Matchmaker Exchange. Disease definitions also vary around the world. Dozens of terminologies and disease registries exist. These are often not included in clinical terminology, such as the International Classification of Diseases, or ICD, which are commonly used in electronic health records. Fundamentally, the definition of a rare disease and how to model it computationally has remained more of an art than a science. So the promise of precision medicine is really, if we think about it, the fact that every individual person is their own N of one disease. And the question is, what are meaningful groupings of those diseases? And so we think about the disease state as being the phenotypic outcome that is the result of one's genetic endowment within an environment or series of environments over time. And the question is, what signs and symptoms, demographics, exposures, diet, traits, et cetera, help us classify patients into those meaningful groups that allow us to perform diagnostics or development treatment strategies. So the prevailing clinical diagnostic pipelines leverage really only a tiny fraction of the data available. So over on the left, we have our candidate patient with a candidate disease. And fundamentally, the goal is to compare this individual against reference information at the population or cohort level. And in genomic diagnostics, that's largely comparing a panel or a whole exome or whole genome to a genomic reference standard. Oftentimes, those genomic reference standards are not themselves very representative of individuals from around the world. And then furthermore, we fundamentally don't often take advantage of clinical phenotypes, multi-omic phenotypes, socioeconomic factors, and environmental exposure that can also influence a person's disease state. And we also lack for reference information about many of these resources, such as disease definitions, population frequencies, correlations with disease, population-based statistics, and then information about exposures from pathogen surveillance, survey instruments, or GIS or other kind of environmental data. And so fundamentally, we have this huge underutilization of data that really prevents us from diagnosing as many patients as possible that have rare genetic diseases. So the human phenotype ontology is a special kind of terminology in the sense that it's similar to the International Classification of Diseases, but it exists as a multiple inheritance graph. So you can see that individual concepts in this graph, such as deeply set I, are classified in multiple ways. And this allows us to have a much richer representation of the biology of the features of an individual patient. And these phenotyping terms are over 17,000 terms now, allow us to build computational disease models that we use for our diagnostic applications, which I'll show you in a minute. The HPO is widely adopted in many rare disease genomic diagnostic tools, such as the 100,000 Genomes Project, SolveRD. It's used by the NIH Undiagnosed Diseases Program and Network, and many other commercial and public resources. Also, what makes the HPO different is its logical interoperability with basic research data. So, for example, the term on the left, hyposmia, is actually represented as a computational axiom relating the gene ontology term, the sensory perception of smell, or rather its lack, which has been annotated to over 34,000 different times to different genes in 22 different species. And so this huge amount of basic science data then can be used together when we're performing semantic phenotype on patients in the clinic. So this is what that phenotype-based diagnosis looks like. So here we have a patient on the left, a 3-year-old girl, on the right, a 14-year-old boy, both come into the same clinic. And within a few weeks of each other, standard panels were not able to identify what ended up being two different variants in the same gene, KMT2A, which demonstrated that these two patients had different variations on Wiedemann-Steiner syndrome. And you can see here at the top, for example, that the 14-year-old boy had a long toe. This is not a term that you necessarily would find in a normal electronic health record because it's really representing the biology of that patient. But it is really important for our diagnostic use case here, where we can compare that to our gold standard for Wiedemann-Steiner, where there are short toes, and those are clearly opposite phenotypes, but they are obviously related to one another. In other cases, we might have exactly the same match. For example, we might have delayed speech and language development. For our little girl on the right, we might have an exact match for microcephaly, whereas for our 14-year-old boy, it's macrocephaly. But fundamentally, this is a computational challenge of finding the closest match for a set of terms in the graph to our graph that we have created as a gold standard for all of the for over 7,500 rare diseases. So for diagnosis to work like this, however, the reference knowledge for each disease needs to be systematically queryable. And assembling the source of disease definitions and annotations is very, very hard. So what is the most clinically useful way to define and group diseases? We have terminologies that represent complex diseases, cancer, infectious diseases, rare diseases, Mendelian diseases, and there really are very, very many disease terminologies and resources. But we needed a way to represent the structures across all these different categories and a systematic way of relating them in a computational manner. So why not just use mappings across these different terminologies? So these can absolutely be used to crosswalk across, and many people do, in fact, do that, where they map SNOMED to ICD, for example. But the problems are that these are mutually inconsistent oftentimes, that there's n squared minus n sets of mappings, and that it's really hard to figure out what actually constitutes a one-to-one equivalency. And this really means that different terminologies used in different contexts for diagnostics work are going to provide different answers in the diagnostic context, meaning that not every patient gets the maximal potential opportunities for a diagnosis because of this scatter in terms of disease representation knowledge. So here's an example. These are the same related sets of concepts in 3 different terminologies coming from Orphanet, the National Cancer Institute's Thesaurus, and the Disease Ontology. Here on the left, you can see there's peroxisome biogenesis disorder. It's a subclass of a rare hereditary metabolic disease with peripheral neuropathy, as well as a subclass of or subtype of leukodystrophy and peroxisomal disease, and you can see that Zellweger syndrome is a subtype of peroxisome biogenesis disorder. However, in the National Cancer Institute's thesaurus, Zellweger syndrome is a subtype of leukodystrophy directly, and is also a subclass of rare non-neoplastic disorders. Furthermore, we can see that Refsum disease is a subtype of Zellweger syndrome, which is really quite different than what we see in Orphanet. And then when you look at the disease ontology, we see similarly Zellweger is a subtype directly of peroxisomal disease, but has infantile Refsum disease as a sibling. And so these are very different structures representing the same concepts. Where is, the correct what is the correct representation? We have different labels, different parents, different children, different synonyms, and different text definitions. And so figuring out how to detangle this kind of difficult structures that don't align across these different sources is really fundamental to building a good diagnostic pipeline. So mapping alone is not going to solve our problem. It's what we attach to the disease classes that really matters. And the fact of the matter is that different communities annotate different relationships at different levels of granularity and using different vocabularies. So those were just the disease entities on that prior slide. When we actually look at what's attached to those disease entities, such as variants, genes, phenotypic features, treatments, environmental exposures, or drugs, or other toxins, different communities are annotating different things. So, for example, ClinVar associates diseases to variants, whereas Orphanet associates diseases to phenotypes as well as to genes. And so we fundamentally want to try to align and integrate these different important resources to give us the most complete picture of what a disease definition actually is. So we created an evidence-based curated merging of equivalent disease concepts from across all of these different, very different many resources over on the left using an algorithm called KBOOM, the Bayesian owl ontology merging algorithm, which uses logical and probabilistic inference to determine what concepts are actually equivalent. And fundamentally, you can think about this as just looking at all of the different labels and cross-references and synonyms and the structure of the graph to try to figure out what might be the most similar set of concepts that we would call an equivalence clique, which is essentially identical concepts. And what the algorithm does is it provides a probability score that shows us things that are likely that are basically in conflict. And what we found is that a lot of the upstream resources have many different errors in them. So, for example, we might have diabetes type 2 with the Roman numeral 2 and diabetes type 2 with the alphanumeric number 2 coming from the same resource, which is not possible, but it's an error in the actual source terminology. And so that's something that we can curate and feed back to those resources, which actually helps those communities' resources improve over time as well. And this results in the terminology which we call Mondo, which means for the world. And the idea is that Mondo captures the full provenance of all of those equivalencies across many different resources, allowing one to use it sort of as air traffic control across all the different knowledge resources and countries. And so this is just an example for that same concept that was in the original example for adult Refsum disease. So we have a Mondo identifier. And we can see in the center of the graph here that there are term identifiers for the synonym source for each of those terms. So adult Refsum disease would have that OMIM number in pink, that UMLS number in green, et cetera, through the central column. So that's the equivalence clique. So all these different concepts from the different terminologies are considered equivalent. And we can point at what their source synonyms are there in column A. So not all synonyms are found in all sources, and not all synonyms are necessarily considered equivalent in all contexts. And you can see on the right some examples of that, where we have some either unscoped cross references or synonyms, or some broader to narrower types of synonyms, for example, to phytanic acid storage disease, which is not exactly the same as adult Refsum disease, but is a related concept. And so this really helps us computationally disentangle the different associations into the Mondo terminology, which now can be used as the handle by which we can perform our disease diagnostics that I showed at the outset. So by doing this, we were actually able to count the number of rare diseases. And this number is not something to be quoted. It is purely an estimate. And what we did was we actually looked at, how many rare diseases do we actually find if we perform this Mondo merger? And we found over 10,443 unique rare disease concepts. We actually believe the estimate to be over 12,000. Where those prior estimates I mentioned earlier were only 7,500. But what's important here is not actually the exact number. It's the fact that many diseases exist in only one source. And so you can see there that bar on the left, where we have around 1,600 diseases that were found only in orphanet. Similarly, we have many diseases that are found only in OMIM, around 1,480. And then even more importantly is when we look at just these 5 resources, the NCIT, the Disease Ontology, NIH's GARD, Orphanet, and OMIM, we found that only 333 shared disease concepts existed in all 5 sources. So this really, really tells us that we have to be integrating these different knowledge sources if we're ever going to have a hope of using these different knowledge sources in a computationally comprehensive manner for rare disease diagnostics. And I should also point out that just recently we had a large community initiative from around the world basically agree to now reference over 10,000 known rare diseases moving forward, including the National Institutes of Health here in the United States. So combining genomic and phenomic data improves variant prioritization for diagnosis. So in the middle here, we see the patient, and I've just been talking about the HPO-based patient phenotypes shown in green. We can compare those phenotypes to all human diseases and causal genes, as I showed you earlier with the Weidemann-Steiner example. But we can also use protein-protein interactions for a guilt by association, as well as mouse and fish orthologs and phenotypes to try to expand what our knowledge might be about orthologs in those other species. And by using this phenotype-based phenotypic similarity, we can combine that with more standard variant reference data when we compare the patient's candidate variants using standard methods such as removing off-target variants, common variants, benign variants, and variants that are inconsistent with the mode of inheritance, as well as pathogenicity determinations. And by doing that together, we have a much better efficacy rate for rare disease diagnostics. And I should mention that this is all available in the tool called Exomizer, which also analyzes whole genomes. We were able to evaluate Exomizer in the 100,000 Genomes Project, where 2,183 proband with 161 undiagnosed disorders were evaluated. And we found that 25% of probands received a genetic diagnosis, with 86% of the diagnoses identified through an automated pipeline using Exomizer. And importantly, 25% of the genetic diagnoses had immediate ramifications for clinical decision-making. So what this tells us is that by leveraging more data, the phenotypic data, the model organism data, the protein-protein interaction data, that we're able to improve the diagnostic yield and the clinical outcomes for these rare disease patients. So I wanted to move on a little bit to talk about rare disease incidence and incidence and prevalence. And so we performed an evaluation across several healthcare systems to try to understand, what do we understand about the prevalence, and in particular, we would really like to know more about the expressivity and penetrance of the individual phenotypic features of these diseases. So this was just a first effort to really think about how well are we actually evaluating even just the prevalence, let alone the sort of variability within a given disease. So here we took 14 rare diseases shown on the left and looked at the prevalence in our health system at Oregon Health and Science University at the time. And we calculated prevalence of these rare diseases. Now, these are rare diseases that do have an ICD code. And we compared them to the published estimate of the population prevalence. And you can see that those numbers are really quite different. 0.015 versus 0.009, 0.012 versus 0.005, et cetera. And in some cases, really quite different. We also looked across other healthcare systems, Sanford, Eversana, and at NIH, as well as comparing to the medical literature. And we found that these percentages varied quite a lot. And you can see, just looking across these different diseases, that we really see quite a lot of differences. Now, there are many reasons why this might be. It could be due to the rarity of these diseases, and that some some patients with these diseases go to certain healthcare systems more than others, for example, to NIH, that the coding of these rare diseases is different in the different systems. There are also likely to be insurance biases, because in some places, the coding of a rare disease may preclude care. And so we know that these problems create some of the heterogeneity, but the point being that we are really challenged to actually calculate the exact prevalence of rare diseases across different systems. We also found that the age at the first diagnosis varies by rare disease. And there's a large spread of initial diagnoses, both due to challenges in diagnoses, but also challenges in obtaining correct information from the electronic health record. So this is what this looks like for an individual patient here with Batten disease. And this is a patient who is male with the age of diagnosis being 14 years old. And we wanted to evaluate the sort of costs of a diagnosis, or the costs before and after a diagnosis. And so you can kind of see the trajectory here in terms of dollars on the y-axis and time on the x-axis. And you can see different treatments, different development of specific phenotypic features like speech and language issues, epilepsy, and then you can see that diagnosis in the middle. And then we start to see some actual changes in the care regime, where we have an inpatient stay, we have specific drug therapies, and there are a variety of different therapeutics that have been leveraged in that future state. And in this case, we see that the cost has actually gone up. In other cases, we've seen that the cost goes down. And fundamentally, given the variability across health systems, we actually don't know a lot about the overall costs. In some countries, it seems that the economics are such that costs actually go down with the rare disease diagnosis. But this is something we really want to understand if we're going to incentivize providers to code the diseases properly and to improve diagnosis earlier on. So what about all the rare diseases without an ICD code? So we need to get those rare disease definitions into electronic health record, but we have to start where we are. So it's really a Mad Max sitch. We have the International Classification of Diseases at the top, which is a standard hierarchical representation. And we have Mondo that I showed you is a multiple inheritance logical model for how we represent diseases. And we've been working with the WHO to come up with a strategy to actually combine Mondo with ICD-11 in a much more standardized way, so we can take advantage of ICD-11's really excellent upper levels of classification, but Mondo's reconciliation across all the disease knowledge that I showed you earlier. So just a quick example of some of the work now that we're doing with Fanconi anemia, which does not have an ICD code. This is a autosomal recessive genetic disorder. There are more than 20 genes that are involved. We have a representation of this disease in the Monarch Initiative website shown at the link below, and you can see there are many, many different associated phenotypes. And unfortunately, for patients with Fanconi anemia, many children do not get a diagnosis until they're 8 to 12 years old, at which time they might be experiencing bone marrow failure, and then later quite a lot of head and neck cancer. And so we really would like to get these patients diagnosed earlier, and in many cases they can be diagnosed very early in childhood. So we started to work on how we might map rare diseases to enable the semantic comparisons that would allow us to diagnose directly out of the EHR rather than using the HPO annotations that a clinician might create themselves. And so here we may have a suspected Fanconi anemia patient on the left with some concepts that come from SNOMED there in green. We map those to the HPO in the middle in gold and take those phenotypes and then try to compare them against all known diseases, including Fanconi anemia. It's actually a little bit more complicated than that because we want to use the laboratory values. For example, if there's platelets in the plasma by an automated count, those values for that laboratory could be mapped to thrombocytopenia. Similarly, we have notes within the clinical notes that we can actually use named entity recognition or NLP to help extract that information. And so basically trying to get as much information out of the electronic health record as possible to come up with that gold colored disease phenotypic profile on the right that we can use to compare against known rare disease profiles that we've created. So this is the result of having done that. So using HPO-based semantic similarity, we can actually identify patients that may actually have Fanconi anemia in large electronic health record data sets. And this work was performed in the National COVID Cohort Collaborative, which is now the largest U.S. national publicly available HIPAA-limited electronic health record data set. And here you can see in orange Fanconi anemia patients, and then in blue patients with aplastic anemia, and then in gray patients that have similarity to those Fanconi anemia patients that should be further evaluated for Fanconi anemia. And this is looking at approximately 20 million patients. So the takeaways are that we need to code and model rare diseases and their attributes in the same way around the world, that semantic engineering and ontology standards can facilitate interoperability and community alignment, that Mondo and HPO are globally recognized standards for encoding rare disease phenotypes and diseases, and that good disease mappings and open science will save lives. I'd urge you to read our classification, ontology, and precision medicine paper in the New England Journal of Medicine, which really talks about the future of precision medicine and how we can go from our currently discrete clinical data capture that we use in electronic health records, and using semantics can really integrate and include in our rare disease and genetic diagnostics emerging high throughput data coming from pedigrees, wearables, biomonitoring, microbiomes, and all of the other omics. And with that, I'd just like to say thanks to a very, very large number of people, and especially to all of the data sources that provide the Monarch Initiative our data in order to help improve the lives of patients with rare diseases. Thanks very much.
So although Dr. Haendel is not present today, we would still like to express our thanks for her excellent presentation and her willingness to participate in the Genetic Summit. We would have loved to have her available, but things do arise and we adjust, as you can tell today. So due to the nature of this presentation, as you can imagine, we, as the MSRG and management team and Genetic Summit team, cannot directly answer her questions related to her presentation. We do, however, invite the audience to reflect on possible implications and applications of this sort of big data and invite a conversation about this topic for the Q&A session. So please continue to place your comments and reflections into the Q&A tab within the attendee website. We have received a number of questions and comments, and I will review those quickly. We cannot certainly answer some of those, but some we will certainly try to do so. The first question we had asked about how this project related to the Mendelian project going on at Baylor and Johns Hopkins. I don't know the answer to that question. Another question asked about does ACMG have an official recommendation regarding buccal samples that are collected in clinic versus allowing home collection. I think that's sort of unrelated to this presentation. That being said, as a clinician, I have not heard of any specific recommendations by the American College, and I can certainly tell you that we do a ton of home collection in our clinic. So we might be doing that wrong, but you can also always reference the ACMG website for their official statements as those are posted. Another question came that says, how does Mondo compare with HPO terms? From what I can gather, I believe Mondo includes the HPO terms as part of one of its many components. So let me just see. Okay. Had way too many here. So
there's one more, Jan. Got it?
There was a comment, and this was a comment and not necessarily a question, that said it would be great if there were a patient-facing website for families of children with undiagnosed disease to help them ask their care team to enter their child's symptoms using all the different ontologies. There was another response to that, which unfortunately which sort of made a link to the Matchmaker exchange, which is available for clinicians to sort of, when we have a unique particular genetic finding, that we can put it in that and connect with other individuals sort of across the country and across the world. I would have to say, from a clinical standpoint, my response to that would be, oh my gosh, what a lot of work. There might be better take up, I think, when someone's asking to put a lot of particular clinical information into a database, that some of the newer approaches is actually having families put that data in. Families, of course, have a kind of a very specific intent for care of their children. Not that the providers don't, but sometimes these patient-driven databases actually get good data from the families, more complete data. And again, I have to say, care providers don't always have that time unless we had a designated data entry person, which most of us sadly do not. And finally, there was a quick question about when ICD-11 came to play. The team quickly Googled that answer because, again, it wasn't part of this presentation. It looks like it actually opened up in 2019 and was implemented in mostly January of 2022. And there's already talk of ICD-12 being released as well. So it looks like it's probably several years before full ICD-11 implementation, but undoubtedly, as soon as we get that done, we'll be on to the next version of that. So at any rate, I would like to actually open it up to our management team and our genetics summit team to see and hear if they have comments about Dr. Haendel's presentation and what their takeaway points might well have been. Kathy, would you start us off?
Yeah, sure, Jan, I'd be happy to. And I'll have some really brief comments. I think I myself haven't been involved in surveillance work with the CDC and various projects in cataloging known but relatively uncommon conditions like sickle cell disease. And it is striking the effort that is needed and is ongoing with Dr. Haendel's work and others to really get common common terminology. I mean, it's just so important. You know, I'm not sure there's an ICD-11 code for a long toe, as she gave an example, right? And the challenge comes is that any given practitioner may see a unique, probably syndromic, or there are clues in the spectrum of manifestations in a given individual, but given the rarity of a condition, they may never see another in their practice, or maybe even in the group of practices with which they're affiliated. So it really does take surfing the data and what is hopefully consistently characterized clinical phenotypic information and even laboratory data to try and pull together a diagnostic, a new diagnosis, recognize a truly rare condition. No one practice, no one provider, even when consistently putting in symptoms, is going to see enough of those individuals to characterize the pattern. So I think it's really important work, especially in the rare diseases space. And I can just say from sickle cell, which is at least a more of a known entity, it's a mess with characterization and inconsistent use of ICD-10 codes. We need to go beyond just what the coding looks like, which is really there for billing purposes. And it's this kind of work, I think, that gives hope with a lot of biostatistical evaluation that we're going to start to recognize conditions.
Thank you, Kathy. I think I would add to that too, in that I think once we identify a condition, we don't really know the spectrum. We know the classic presentation, and speaking largely from the standpoint of metabolic disorders, you know, we know the classic presentation because we had those diagnoses biochemically well before we started doing newborn screening. Newborn screening for many of the metabolic disorders opened up a broad spectrum of disease phenotype, from really, particularly on the milder end of the spectrum. And, you know, half of us would probably have missed those kids in clinic. So again, it goes back to kind of, you know, making accurate diagnoses and seeing that broad spectrum, which is really hard to do when you may see only one or two of those kids in your career per se. And so having someplace like Mondo, where this information is gathered, and particularly combining the phenotype and genotype information, because some of these diagnoses wouldn't even be made clinically. They're being made only when we do genetic testing because of a newborn screen, for example, that biochemically they have such mild findings they maybe never present. But so it's kind of two parts of the spectrum, you know, getting the correct diagnosis, but recognizing the broad spectrum of disease as well. So, Liza, how about your thoughts?
Sorry, took me a second to find mute. Yeah, this is great. So this is an interesting presentation. And before I heard it, I really didn't know much about this kind of work that was happening. I think, you know, better, more consistent and more comprehensive definitions and the combining of data sources really have the potential to improve our understanding of the epidemiology of rare diseases in both the US and worldwide. I think it's good, right? That's a good thing because it helps us understand the potential impact of a disease or a class of diseases, but also for establishing research programs and supporting delivery models that serve people and that support people with those disorders. So that's key, of course, for clinicians, for advocates, for families, and for individuals. I think this presentation really kind of reinforces some disparities that exist in access to care, potentially. You know, it reminds me how unfortunate it is that administrative systems drive or influence insurance coverage or potential payment for care or services. You know, the definition's not there, so we can't bill for this. And that has major impacts for families. So it's really encouraging to see this kind of work happening, these systems interacting, people thinking about how to make them or facilitate them interacting. It's important for for clinical purposes, and it's important for the people who are affected. Finally, I'll just kind of say, I come at this from sort of a policy research lens, and, policymakers don't fund research on or programs for things that they don't know exist. And more data is absolutely better. I kind of made a similar comment yesterday in my talk, but there are also limited resources, and better data doesn't always lead to more investment, particularly when there are many different competing priorities. But more information does help advocates, community leaders, and others think about how policy or other decisions might be useful for better understanding or improving care for a particular disorder or for a group of disorders.
Thank you, Liza. That was excellent. And we always appreciate the lens that you look at the world is different than ours, and that you come from a policy and stats standpoint versus a very clinical standpoint. So thank you. That brings in new insight as well. And Christy, you always provide us with kind of the consumer and advocate lens as well. Your thoughts on today's presentation?
Yeah, thanks, Jan. I got to watch this presentation a couple months ago, a similar one, and brought it to the planning committee. So I would just reference that for everybody who would like to learn more about this. This was done at NHGRI a couple months back with some more insight and some other speakers. So I'll point everybody's attention to that as well. But I was really struck when I first heard about this because as our family experienced, our child's phenotype changed with a lot of environmental factors. And so I was really excited to see that one of those first slides where it's looking at the phenotype, the genotype, and environmental pieces as well. And it was in a presentation that I did with Dr. Kaler at our 2021 Genetics Summit, where he really made me realize that there are genetic conditions that do change with environment, PKU being one of them, with an environmental diet treatment. And so just thinking differently about the whole world of genetics and genomics and phenotypes and genotypes, and really bringing all these ideas together was really exciting for me as a family, because I remember asking some of these questions on our genetic journey, like, what about this? What about this? And those things that were important to me as a mom that I was seeing in my own house maybe didn't quite make it into the right place in the electronic medical record to be captured, or on the one line that the physician could fill out for the phenotype that went along with the exome sequence order. And so to me, having a place for all of this to come together, though it may not be perfect, it's something to bring all of these sources together. It looks super complicated, I think, is my first initial knee-jerk reaction, and love the suggestion of how can families be part of this and in encouraging their providers. But I love your perspective, Jan, because I do think it might need to come from the families. I remember seeing the order for my daughter's whole exome sequencing and wondering why they hadn't written certain things down, but then realizing maybe I should have suggested they wrote a couple things down that I thought were odd in our that were very specific phenotypes for her journey. So I do think it's a partnership, like we saw with Leah and Maria's presentation earlier today, is that this is really a place where physicians and researchers and families can all come together and contribute.
Thank you, Christy. Yeah, I would comment that, from a very clinical standpoint, very frequently when we send exome or genome sequencing for a child in the hospital, we'll send some clinical information and then we'll get a preliminary report back. And very frequently the lab asks us, what new information have you gotten on this baby, this child, since you sent the sample? And it will help in the interpretation of how they look at the results. Are they clinically relevant? Do they make sense? Do they help explain this child? So yeah, it's always nice the more information you can provide from an interpretation standpoint. We had a couple additional comments. We mentioned, family involvement, but there's a comment here. It says, it's interesting that many rare diseases are listed only in one database. Are there ways for genetic counselors to help get conditions listed in all of the different databases for improving accessibility of information? I have no idea about that, but I do think, again, from a practical standpoint, the more information you can provide clinically to help interpret results, the better. But I don't know how we would have necessarily access to add information to databases. There is in the back with lab interpretation and the labs, when they get variants, sending them to ClinVar and those sorts of things, that information's added. I don't know if GCs could have a role in some of that. I have no idea. Another individual offered that phenomenizer, if I said that right, phenomenizer is another good one, data set one for putting in a collection of symptoms. So there are other databases out there that you can add information to. Another comment was thinking of how phenotypes may be coded differently in different systems. Could this quote behind the scenes coding data eventually be part of the medical record that a patient has a right to access for transparency? I was told it was inaccessible to me. I think that's a great idea. And certainly I think, there's a lot of push in electronic medical records to code things similarly and use the link codes and those sorts of things so that they all interact. But it would be wonderful if we could have electronic medical records that pulled this different information and then, helped send it to where it needed to go for aiding in interpretation and diagnosis. So at this point, we are actually at time. So again, we want to thank Dr. Haendel for her presentation.
For more information, visit mountainstatesgenetics.org/contact. This activity was brought to you by Mountain States Regional Genetics Network. Please enter this code in the evaluation survey in order to claim genetic counseling, CEU GS23-12.